So thank you, Ollie, for the introduction, and I'm pleased to be here to talk about this. So in COVID, this whole pandemic, we've been talking so much about the spike protein, so much about antibodies, but there's a whole aspect of T cells which exists in the immune system that I believe is rather underappreciated in this whole pandemic. So today's talk here, I'm going to be sharing some of the insights we have learned from our routine work. So we've developed a test and used it for routine purposes, and also in research. So I'll come into that later. So a brief introduction, a very simple introduction into the immune system and how it reacts to an infection. So here we have an infection of the virus into the epithelial tissue, typically the lungs then, of course. And when an infection occurs, what happens is that there are, of course, the original barriers or original protectors, so the innate immune cells that are there to try to digest, break down the virus, contain the infection. But however, when this clearance or protection is not sufficient, these frontliners will then return back to the lymph nodes, so circulate into the lymph nodes. Think of them going to the base camps of the military and ask for recruitment. And that's where we have this, for example, helper T cells or CD4 T cells. Now on, I'll always be calling them helper T cells. And why are they called helper T cells? Well, their function there is to typically produce cytokines. So these are biochemical mediators that helps activate B cells and also enhance your antibody production and initiate this class switching or affinity maturation. So what that means is that when a B cell produces antibodies, they can even enhance their binding affinity to an antigen. So yeah, antibodies, we know about that, and the purpose of an antibody is really just to bind the virus in the first place and prevent infection from happening. Simultaneously, CD4, so helper T cells, also activate cytotoxic or CD8 T cells. And what these do, you can think of them as a SWAT team, so the special forces that come in and have a tactical approach to kill and eliminate just infected cells. So current diagnostics, of course, focus so much on the RT-PCR for the virus itself that is useful for the acute phase, and then for the antibodies to spike 1 in later phases to show for a response. But what about the whole cellular aspect? You know, I've mentioned that there's this whole aspect of the whole adaptive arm of the immune system, and not many people talk about testing this. So is it possible? Yes, and I will show you briefly how we are able to detect helper T cells and also cytotoxic T cells. So before I go into that, though, I just want to talk about the structure of the virus, because this informs how we can develop tests. Spike has been in the news all the time, but the virus is not just made of the spike protein. There's, of course, other structural components such as the nucleocapsid and the membrane, and other bits as well, which the immune system actually recognizes. So the immune system's response is rather broad. It recognizes multiple aspects of the virus. Spike protein is, of course, critical because it is the protein that helps the entry of the cell into the host, into the humans. So our test, what is our test actually? So here I show you what are the testing conditions we have and how it starts. So we isolate 30 milliliters of whole blood from patients, and we isolate specifically pure peripheral blood mononuclear cells through a gradient centrifugation, and then we culture these cells together with different stimuli derived from the virus. So we use whole proteins, so spike 1 and spike 2 protein, and also the nucleocapsid protein to detect for helper T cells, and also peptide mixes. So what are peptides? That are broken down pieces of the protein. So think of a big protein, it's chopped up into multiple pieces, and this is what is used. We use different mixes to then detect cytotoxic T cells. So 5 days of culture of patient-derived blood cells, peripheral blood mononuclear cells with the stimuli. And at the end of that, we assess by flow cytometry for cell activation, so cell activation markers. I don't want to go into so much detail on how we develop the assay. I rather want to go into specific interesting cases. If you want to read more, you can, of course, scan this code, but we published this data to show how it's validated, and we show a high sensitivity and specificity of this test. And what I also want to stress is that we have used this over the last 2 years, 1.5 years, for routine diagnostics. And here's an example report that we sent out, and we produced this report in French, German, English, and Italian as well. Yeah, you can read more about that here in our webpage. So we developed this test prior to actually the vaccination. We know the vaccination does induce a strong antibody response, that is clear, but what about the T cells? So here I just want to very quickly go through it. Yes, basically, in the beginning, in these naive individuals, they had no T cell responses. After the first dose, you start to generate some already, and there's still presence there. I wouldn't say it's such a boost, but there's a maintenance of the T cell response. In previously infected individuals, we already see that the T cells are there even before the first vaccination, which is clear that they have been immunized by the virus. So yes, T cell responses are induced by vaccination and also past infection. Good, so those are pretty basic things, I would say. Now I want to go into more special situations where this test could be useful. So of course, the first one is here in situations where people are immunocompromised. So either by treatment, by immunodeficiencies, so maybe think about transplantation, where you give your patient a medication to dampen down the inflammation, to prevent rejection of the transplant, or in some cancer patients, particularly blood cancers, or if people are just genetically having an immunodeficiency in the first place, so primary immunodeficiencies. So this is the first case I will show later. So you always see a 1 at the top right later if I'm talking about that. And number 2, does prior vaccination or infection actually still prevent, or rather recognize, the mutant variants? Pierre has shown that how the antibodies may be not as effective to prevent infection in the first place. That does not surprise me, and I'll come to that later. So the first one, immunocompromised individuals. So here, what I wanted to study is if in immunocompromised individuals, could they still have a T cell response to vaccination or to past infection? Here I'm showing you first the background level of people who were never exposed, so not vaccinated, not infected. We don't really see an activation of T cells yet. This makes sense. And the study group that we had in our routine work was over 40 people who had immunosuppression, and then those that had vaccination were 32, and infected was 8, at least in our studies. So here I want to show you that in general, so these are immunosuppressed individuals now. These ones here for helper T cells, here for cytotoxic T cells. In general, you see that there is a upregulation of T cell responses at both the helper T cells and cytotoxic T cell levels, which is great, and that's really reassuring that vaccination can induce this effect. There is also the same trend with infection. I believe it's more a statistical effect of sample size at the moment here. But with that said, though, look at the range here, and I also want to highlight the scale. This is an SI of 10 and up to 100, and yet here there are some that have really no responses. So there are certain immunocompromised individuals that have really good reactions, really good T cell responses, and some that do not. So actually, I would say that for these individuals, while it is reassuring that a lot can have a good T cell protection, it is, I think we have to remember what healthcare is about as well. It's coming to deal about individual patients. And I think this is the point of where we have this very specialized test to actually look into the situation. So moving on a little bit more into immunosuppression, we wanted to see in different types of drugs if we could see if a particular drug particularly reduced T cell efficiency. So here we had about 20 patients that had such immunodeficiencies, mostly targeting the B cells, some for T cells, and some general immunosuppressions. We compared them to healthy donors who received 2 times vaccination. So all these individuals received the 2 doses of vaccination. Here's the response in in healthy controls. So you see a nice T cell response. That's what we showed in our publication. And here we start to see how this helper T cell response is and cytotoxic T cell response. Again, it's similar to what I showed you before. Most are able to react, but there are some that just cannot react properly. And this is why it is still a suggestion that we should look at the individual level to see how the responses are. What was interesting for me too is those drugs that target T cells, they still actually are able to respond generally pretty well, but that also depends so much on what drug you're using. So CTLA-4 really blocks activation of T cells. That one there we see a clear inhibition, but the other ones, which are a little bit more specific, they don't target all the T cells. We still see pretty good responses. Good. So that's how I said. We need to test for the T cells, and with this information of the clinician and the patient, we can come together to actually discuss what is the best care and solution to move forward with. Now I move towards Omicron. So yeah, it's master of antibody evasion. Well, look at this. So this is the spike protein, and you see all this red here is all the number of mutations. There's about 60 total mutations in the spike protein, in the whole virus, and half of them are in the spike protein. This is a schematic of the spike protein now, and prior infection or prior vaccination to the wild type strain, you see an antibody response that is able to recognize the spike protein. However, when mutation occurs, that can lead to a three-dimensional change, and therefore, the antibody now, you see this minor change here now causes a conformational change that allows the antibody to not bind so well. And therefore, this is not surprising for me that the antibody responses are not good anymore because the virus is able to evade, in fact, the antibodies. But, of course, there's the other arm of the immune system where the proteins are actually degraded into linear peptides. So this is just unraveled into a single string, for example, and then chopped up into different pieces. And luckily for us, that's what the T cells recognize. And what I want to point out here is that you see that many of these parts are still conserved, actually, despite the mutations. So actually, we believe at a theoretical level, at least, that T cells are able to recognize the mutants. Of course, if the T cell epitopes itself are mutated, that may be problematic, but I come to that later. You've seen this table before already. That's what we use to stimulate in our tests. Here's the mutational profile of Omicron. So 60 total mutations, 34 residing within spike protein, and the majority within the receptor binding domain. So that's where the virus binds to enter the human cells. So we change our spike protein, so we actually test for spike wild type, but also the RBD, so that we're having some antigen that's specific to Omicron. And then also for the cytotoxic T cells, we adapted our peptide mix. So there was actually, in fact, only 6 out of the 28 total peptides that we use for spike protein were affected by the mutation. And actually, if you look at the HLA alleles that are affected, these are not the typically most abundant allele. So HLA-A2 is the most abundant one. So actually suggesting that most of the population will still have an immunity on a T cell level. Here I want to point out just one example. Here, there's 1 point mutation from the G to D, so there's this mutation. But then, again, this is a linear peptide, it's bound onto HLA, the T cells are able to recognize still the vast majority of the peptide. So, in practice, is immunity still effective against Omicron? What did we see? So at the helper T cell level, here are people who were vaccinated and those infected. So these are people only infected from January onwards, and at that time point was about 80% Omicron in Switzerland. And I would overall say that the ability to recognize wild type and Omicron remains the same. Maybe there's slightly reduction in the helper T cells, but remember this is now targeting specifically the RBD. Whereas for the cytotoxic T cell, I would say that there's still a preservation of the response. So actually, I would say at the T cell level at least, that despite the multitudes of mutation, that T cells are still able to recognize the Omicron strain, and maybe it explains the reduced severity that we see despite the boost in the breakthrough infections we saw at the end of the year and start of the year. So key messages I want to say is that immunity does not just rely on only antibodies, despite how the media only talks about antibodies. And if only one arm is working, this can maybe lead to a not proper clearance of the virus and altered dysregulation of the disease. But when there's a working together of the immune system, there's a better clearance and hopefully a milder disease. So remember, the immune system does not exist in a bubble, it exists as a coordinated response. So this type of analysis for T cells exists. We have used it and we use it in routine work. And this is a complicated test, but it can be done. And it's especially used for people who have no prior antibody responses, where we can actually see T cell response. That will come to the long COVID study later in the discussion. So all in all, I just want to say that this test exists. It's providing an unmet diagnostic tool to look at an individual level of patients. And I believe this exists now. We can use it for future mutants and also even other viruses, because we have to learn something from the 2 years of pandemic. There's something we need to learn out of this to be better prepared for the future. So thank you very much, of course, to the whole team, the science team, and of course the lab team at ADR-AC, and all the many blood donors. Thank you very much.